We have joining with us our moderator, Dr. Jennifer Orthman Murphy, who's with us today. Dr. Orthman Murphy is the Assistant Professor of Neurology and Co-Director of the Age Span Fellowship in MS Neuroinflammatory Disorders at the University of Pennsylvania. Dr. Orthman Murphy runs a translational research program studying the role of glial cells in myelin disorders. She also sees patients with multiple sclerosis and leads an undiagnosed white matter disorders clinic where APBD is the third most common disorder diagnosed. Dr. Orthman Murphy is a reviewer of our $1 Million Bike Ride scientific research grant applications. That is what we'll be updating you on in this next session. And she also serves as co-chair of the Foundation's Scientific, Medical, and Advisory Board.
Thank you so much for that introduction and for being a part of this really powerful and exciting 2 days. And I'm just really grateful to start off on an exciting session where we get to talk about the research investment and hope for the future. So we hope you feel inspired by what's going to be presented in this first session. I am honored to be a reviewer for the APBD Research Foundation. This is a pilot grant program that is in a partnership with the Orphan Disease Center at the University of Pennsylvania, where this program actually offers 39 different research grant opportunities focusing on 31 different rare diseases, where APBD is one of them. And as you heard earlier from Jeff, that an enormous amount of money has been raised. It's really congratulations for that. This is an incredible effort, and already 6 institutions and 14 projects. It's just incredibly impressive. So today we're going to highlight the work that represents the investment from this past year for some really exciting studies. And we hope that it generates questions. We're starting a little bit early, so I love that because it gives us a little bit of extra time. So write down your questions so after the presentations you can put them into the chat. It should generate new questions, maybe even collaborations, and hopefully future discussions and conversations. And so first, I just want to introduce all of our panelists for these updates. So we have Dr. Or Kaklon, who's an assistant professor in the Department of Neurology at Hadassah Hebrew University Medical Center. Dr. Rebecca L. Koch is a postdoctoral associate in the Department of Pediatrics at Duke University Medical Center. And Dr. Kara A. Jones, an associate professor of pathology at Duke University Medical School. And so now I would like to let these researchers share their work first.
Great. Okay, so I guess I don't have control over it. Okay, we'll just ask you to push the button whenever I need. So, okay, this work is actually, most importantly, still in progress. So we don't have results yet because the mice are still being dosed as we speak at the moment. However, I can, of course, share our research plan, rationale, and what we are looking for. So in general, we are actually, we have used or we have experiments on single compounds, small molecules, which we discovered as a part of a high-throughput screen, which was published, so I will not elaborate on it here. And we selected 3 of these molecules that for 2 of which were published, GHF-201, sublicensed to GHF, the company which is represented here by the CEO, Yaniv Glazer. And so GHF-201, GHF-205, on which we have some unpublished results, but a lot of them, and Guaiacol, which was published back in 2018 in collaboration with Dr. Ackman. So next slide. Okay, so the specific aims of this study were first to try different grouping and temporal combinations of Guaiacol, GHF-201, and GHF-205. What do I mean by that? Temporal means pre-onset. So at 1 month of age, we start the treatment. Because we didn't know it before. Now, with the publications of Dr. Minasian, we know that actually the polyglucosans even can start at early age, so early stage. So we started 1 month instead of 4 months, which is what we did before. And the other time is post onset at 6 months of age. And the grouping combinations are just, each of the compounds separately and in combinations with others, as you will see in a moment. So we're going to look at reversal of pathology and also to look at new biomarkers, which is, I know what the MDPR and the ODC is very interested in. Brain spatial using spatial mass spec, serum and CSF metabolome, light chain neurofilament, which has become quite common as a biomarker for neurodegeneration in ALS, for instance, and neuronal and astrocytic, I should say, phenotypic features of the cells. Next. Okay. So can you press the cursor, please, one time? Then it will highlight what I'm trying to show. So it's, yes, okay, thank you. So what I wanted to show you is what are the arms, what are the study groups, and what do they mean? So in other words, what comparisons are we making? What null hypotheses are we testing? So the first null hypothesis, highlighted here in magenta, is to test whether post-onset administration of guaiacol and 205, okay, as you can see here, so at 6 months, produces the same effect as post-onset administration of each of the compounds separately, which is arms 13 and 14, okay? So what we're testing here is that, and yes, I should say we use a parameter called the combination index, okay, to tell if the interaction is synergistic, additive, or antagonistic. So there's a mathematical way to test this. So what we are testing is whether the glycogen synthase inhibitors, guaiacol and GHF-205, okay, for guaiacol, we know it. For 205, it's not completely. I mean, we know it also that it inhibits the glycogen synthase, but the target is not completely validated, almost completely. But we know it inhibits glycogen synthase. So, in other words, when these two glycogen synthase inhibitors are administered together, can they synergize, or do they have only an additive effect as each one of them separately? Okay, so there is also a big question mark here, assuming they have a post-onset effect at all, because prior neurodegeneration might be irreparable. We know this because when we did it with GHF-201, didn't have any corrective effect if it was applied post-onset. But we will test this. Number 2, next. So the second comparison is actually the same as the first comparison, but at the pre-onset. Okay, so we're going to test the 205 and guaiacol together and compare the outcome measures to what happens when you apply them, each one of them separately. Next. Okay, so the third comparison is we compare pre-treatment with the polyglucosan degrading compound GHF-201 that you can see here, so at the age of 1 month, followed by a post-onset inhibition of glycogen synthesis, okay, by guaiacol, for instance. So we will ask if this treatment, so degradation pre-onset followed by post-onset inhibition of glycogen synthase, has the same therapeutic effect as the post-onset inhibition alone. And we expect to reject this null hypothesis. We expect that it will the treatment, the effect will be different, because we expect the pre-onset degradation to amplify the effect of glycogen synthase inhibitor, or even to enable its effect. Because, as I said, post-onset, the effect is very attenuated. Next. So the next comparison we are making is we pre-treat with the GC inhibitor Guaiacol, as you can see here, at the age of 1 month, followed by GHF21 mediated degradation. Okay. This arm, okay, this study arm is compared to post-onset, as you can see here in arm number 12, post-onset polyglucosan degradation alone. Okay, so this null hypothesis is also expected to be rejected because post-onset treatment is not therapeutic. Okay, and while Guaiacol preventative treatment was, this was published in APBD. Okay, I should also say another potential reason for rejecting this null hypothesis is that we are now introducing Guaiacol as a new nanodroplet formulation, which can reach the brain as opposed to what we did previously when Guaiacol was administered in drinking water and was not able to reach the brain. We did the pharmacokinetics. So this expected brain biodistribution is expected to enhance the effect of, or amplify the effect of the therapeutic effect in general. Okay, so when you when the starting point is with less glycogen, possibly GHF21 applied post-onset may be more effective or attenuate prior neurodegeneration. Next. So the next comparison, shown here in brown, is the pre-onset administration of GHF205, okay, the age of 1 month, GHF21, GHF205, and Guaiacol, all three of them. And as you can see, you can compare this to the effect of each one of these separately, okay, arms 9, 10, and 11. Okay, so this actually, if this null hypothesis is rejected, it will demonstrate a synergistic or additive effect, and we will calculate it using the combination index as I explained. And the last comparison, next. Okay, it's the same as the previous one, but we'll do post-onset administration, not pre-onset, but post-onset administration of each compound alone, and compare it to all of them together. So we will also compare, because we have this information, all the preventative treatments and their matching post-onset treatments. So, for instance, we will prepare a pre-onset 205 and Guaiacol with post-onset 205 and Guaiacol. Okay, and then we can test actually if there is a preventative inhibition of either glycogen synthesis or enhancement of polyglucosan degradation is more effective than its post-onset inhibition, which is expected. Okay, because we demonstrated this for GHF201. Okay, next. Okay, so what are we going to test? We're going to test the effects of the compound combinations on the basic parameters, locomotor parameters, such as extension reflex, writing reflex. By the way, extension reflex is very interesting because it's related to pyramidal signs, and we already demonstrated that there is a change in extension reflex. So this is was very encouraging. We did it already 3 years ago. It was published, and the grip strength. Okay, so locomotor parameters. Then we're going to look at nociceptive parameters, and this is actually interesting because we got reports from patients after the treatment is, the treatment with GHF21 is in compassionate use, and we got some reports that the sensation in the feet was regained after decades where it was lost, and therefore we wanted to test it. We already have results for 205. So we're going to test thermal pain sensation, cold sensation, and sensory motor deficiency, sticky tape, how fast the mice can remove sticky tape from their paws. What is interesting that I want to point out is what is called the Miles test, which because you have to tease apart the locomotion deficiency from the sensational deficiency. For instance, maybe the mice cannot lick their paws fast enough after being exposed to thermal pain, just because of time's up, so we'll try to hurry up. Just because of the motor deficiency. So this is, and in order to do it, we actually look at the neurogenic inflammation test instead. Then we're going to look, so I'm going to go quickly over the other things, at polyglucosan body accumulation. No, please go back. Polyglucosan body accumulation, astrogliosis and fibrosis, and blood glucose in blood biochemistry. So I don't have much time, but I just wanted to highlight some of the new biomarkers. So we're going to look at spatial metabolomic, untargeted metabolomic, which areas of the brains are affected by a new technique that is not now available to us called the DESI-MSI. And lastly, can you go forward? We're going to integrate all this data. So first of all, we will isolate neurons and astrocytes. It's possible to do it using a MAX technology, and then look at the cell feature, mitochondrial, lysosomal, how they look. And then we're going to use a method, a statistical method called unsupervised k-means analysis, and see if these cell features actually can cluster with the metabolic feature and can classify the different arms together. So can determine how much they contribute to the entire heterogeneity. And what would be interesting is this last thing here, is that when we integrate the metabolic pathways and the phenotypic data with concomitant compounds, we can see the relationship that we find with the concomitant compound treatment is or is not similar to the same relations without the treatment, so with vehicle. And when we have changes in this relationship, so for instance, if you have more active lysosomes, you have more degradation of polyamines, for instance. This relationship between the organelle and the metabolic, if these relationships are changed, then it can actually suggest that these are new targetable pathways. So this is why it's important to do this integration analysis. So that's about all. Sorry, I went over time, and thank you for listening.
Hi, everyone. Thank you to the APBD Research Foundation for this opportunity. I'm Becca Koch, and I'll be speaking on behalf of the Duke University team for our recently awarded grant, $1 Million Bike Ride. Our study is called Deep Tissue and Cellular Phenotyping of APBD, a bench-to-bedside approach. So in recent years, there has been a growing understanding of APBD on the GSD4 disease spectrum. And so this includes broad neurological manifestations, as well as individuals who have been diagnosed with hepatic GSD4 in childhood that then develop APBD in adulthood. Some children, adolescents, and young adults with GSD4 are now showing early neurologic manifestations that are consistent with what we have historically coined the term APBD. And we know from a healthcare standpoint that multidisciplinary management is required. And so these references on the bottom are just recent ones from our group, but there are several others from other groups that are highlighting this variability and APBD as a spectrum disorder. But despite these recent advancements in our understanding of APBD natural history, our knowledge in the specific pathology as the disease progresses still remains inadequate. And it's critical that we really understand the disease progression of GSD4, including APBD, to not only improve patient management, but to improve the efficacy and the specificity of therapeutic interventions. And so that was really the background and the justification for this work. And so the need for this tissue and cellular phenotyping of APBD is because we can all agree that the CNS, or the central nervous system, is the primary target for therapies treating APBD. However, our understanding of how the disease progresses in the CNS in APBD is limited, and it's limited to functional studies, again, very helpful, but limited in their own way. Imaging, such as brain MRIs, as well as published autopsies. And so there have been 5 published autopsies from confirmed diagnoses. But even within those 5, they're restricted to select areas of the CNS, as well as select tissues. And even so, there are conflicting findings. So it just makes it a bit confusing on how to interpret these results. And we do know that the GBE1 YSYS knock-in mouse model does recapitulate the APDD type from a neurologic standpoint, which is incredibly helpful. But again, I think we could improve on our knowledge on how the disease progresses at the cellular and tissue level, even in that mouse model. And so here is our schematic summary for this study. At the time when we submitted it, we had one patient who had donated his tissues to research. We had one autopsy, but at this point, we now have two. And we're also looking at the mouse model at several time points. And in the middle here, I just listed out our main tissues of interest, but by no means is this comprehensive. This is, again, just a select list for this presentation. And with that, we're analyzing those tissues to understand the tissue and cellular involvement, and also quantifying the level of polyglucosamic accumulation and comparing that degree of accumulation to things like serum biomarkers and other measurable disease outcomes. So as a brief overview, again, I'm not going to get too into each of these autopsies as well as mouse model, but I wanted to give you a brief overview because these are preliminary results, and I look forward to in future years talking about the comprehensive results that comes out of the study. But this is autopsy number 1. This individual died at 47 years of age. I listed his variants there. I did want to note that the two variants in both this case and the other case are not, these individuals were not homozygous for the Y32 variant, and they also do not harbor that intronic variant that we've seen in the Ashkenazi Jewish population. So I just wanted to note that. At 2 years of age, this individual presented with massive hepatomegaly, and on liver biopsy, bridging fibrosis was noted. He was diagnosed with hepatic GSD4 based on deficient GBE activity in skin fibroblasts, and liver disease had stabilized without liver transplantation, so therefore he was labeled as non-progressive hepatic GSD4. However, in his mid-30s, he presented with symptoms consistent with APDD. He received genetic testing, and the diagnosis of APDD was confirmed. And in this case, the cause of death was medical assistance in dying. So here are just representative images from his autopsy tissues. On the left hand, I show images of the hippocampus. You can see this widespread polyglucosan as well as reactive astrocytes. On the right side, because of his history of liver disease, we did take a close look at the liver. And you can see here the liver fibrosis did not regress. In fact, it was still very much visible to the point of cirrhosis in the left lobe. But interestingly, this was not associated with a lot of polyglucosan. Polyglucosan was really not observed to a high level in his liver, despite the liver transaminases being normal and there being no other signs of overt liver disease that would be what we typically see with this degree of fibrosis and cirrhosis. At the second autopsy, this individual died at age 55. Again, his variants are listed there. In his mid-30s is when he presented with symptoms consistent with APDD. But unfortunately, he was misdiagnosed as having multiple sclerosis. But fortunately, 4 years later, he did receive genetic testing, which confirmed the APDD diagnosis. His cause of death was sepsis and hypoxic respiratory failure in the setting of multiple infections, including a UTI and aspiration pneumonia. And I do want to note his family history. He had a younger brother who developed severe liver disease and died at age 2.5 years from complications post-liver transplantation. And this was confirmed to be GSD4. So here are the preliminary results from his autopsy analysis. Again, we see this widespread polyglucosan accumulation in the hippocampus and astrocytes on the left. And then on the right side, I just show select tissues. In top right, you see the polyglucosan throughout the cervical spinal cord. And then on the bottom right, in the small intestine as well. Notably, the liver was donated, so it was not available for evaluation at autopsy. So I cannot comment on the state of his liver. Next, I'm showing images from the mouse model. So this is, again, select regions of the central nervous system over time, showing that polyglucosan accumulation, as well as the spinal cord. And so what I want to show here is this is with PAS diastase staining, more as a direct method of polyglucosan accumulation. And how we're also approaching this, both with the PAS and diastase staining, but we're also using this immunofluorescence staining. This is very helpful in visually showing where the polyglucosan bodies are, but also allowing for co-localization and overlaying of images, because you can do this type of staining with other antibodies. And so this also gives a very clear way to approach this quantitatively. So here at 6 months, this is the size of the particle and area covered by the glycogen synthase staining. So again, indirect method of that polyglucosan accumulation. And again, here you can clearly see the differences. Again, this is very preliminary, but just showing you that this would be another endpoint as we assess treatment efficacy. And you can see the differences between a disease state and non-disease state at 6 months of age. Next, we did a similar approach, but with GFAP as a marker for astrocytes and reactive astrocytes. So here again, the same thing. Interestingly, we are looking at the pattern of astrocyte involvement in this disease. And so here you can see over time how that reactivity is becoming more apparent at 6 months of age versus 9 months or 12 months and earlier stages of the disease. And again, this is with mean intensity. So again, being a way to quantitatively assess this involvement of astrocytes. And now this is just a very brief overview, but one of our next steps is utilize NET glycogen synthase as an indirect method for detecting polyglucosan and looking at how that polyglucosan is localizing using overlaid images. And so here you can see with the involvement of neurons and astrocytes, how and where this polyglucosan is accumulating. So again, just giving us more of an indication of the global multicellular progressive disease pathology. So we'll be doing this for all different age points. And their next steps also include using the same immunofluorescence model on the autopsy tissue samples as well. We'll also be quantifying the polyglucosan accumulation in different regions and determining the degree of polyglucosan accumulation in different cell types. And lastly, we'll be comparing that location and quantity of the polyglucosan to disease outcomes. So in the mouse model, that would be something like biomarkers and functional testing. And in patients, anything that we can glean from the medical records that would be measurable. So I just want to close with my acknowledgements. Of course, thank you very much to the funding source, the $1 Million Bike Ride, for funding this research, the entire Kishnani lab and research team for both their assistance in the bench and the bedside. And of course, our wonderful pathology collaborators, Dr. Kara Jones and Dr. Will Jack from Duke University, who are instrumental in assessing the CNS, muscle, and liver pathology, and also to our colleagues at NYU Langone, as well as Queens Health System, for helping us with these autopsies. And most importantly, I do want to thank the decedents and their families for allowing this important work to happen. Thank you very much.
Thank you. We're now ready to move into Q&A. Jennifer, I'll bring you to the forefront and also our panelists.
So thank you to Dr. Kaplan and Dr. Koch for these exciting talks. 10 minutes is a short amount of time to lay out all this data that you already have in your plans. And I just really appreciate what you were able to share with us so far. I just wanted to share that this type of funding is an initial investment. It's a pilot grant to get these exciting projects started to go towards these future larger funding sources that we're actually going to talk about later in this conference. So to sort of keep that in mind. So now it's time for everybody to enter their questions so that we can look, but I'm going to start it off. I have a lot, so we can definitely fill up the time. I'll try to behave myself. But one of the things that we're also going to talk about later in the conference is the idea of biomarkers. And so I think this is something that you've both addressed, and I'm going to bring this back to Dr. Kaplan so you have a little bit more time to sort of wax on and off about this, because your approach is looking at both pre and post manifestation, which, in the ideal world, we'd like to treat people before they ever develop symptoms and prevent it from happening. But I think your goal of integrating all of your findings into see what markers will change over time to identify novel targets is also really exciting. And so what are the sort of things that we can expect to come out of this? Are these going to be, do you think, the disease-specific markers, like APB-specific biomarkers that will be particular to GSD4, or are these going to be the markers within the field of neurodegeneration where we don't have any treatments yet to fix the brain? For example, our nervous system.
So I can actually focus on maybe two expected biomarkers in the metabolome world. And so this is something which you already know from the patients, okay? Because we did their blood, we know their blood metabolomic signature. So what happens in general is that when you cannot degrade glycogen as a source of energy or carbohydrates, you use lipids instead. And in general, what we see in the patients is there is a decline in lipid levels. So phospholipids, acylcarnitines, fatty acids, in general, drop in the blood because they're being utilized. and we see this is being restored after the treatment. Okay, I have actually lots of data to show. Some of them were shown as preliminary data in the application, but this is definitely restored. So, which means that they can degrade glycogen better. Another really exciting observation that we made, and it was quite reproducible, I have to say, in all patients, is the increase in neurotransmitters, which was really a nice surprise. So we have serotonin, glutamate, and dopamine being increased. And for glutamate and glutamine, actually, I can actually provide an explanation. And now there's also a publication that actually, as expected, when glycogen is degraded, it feeds into the TCA cycle, and then alpha ketoglutarate can actually be shifted towards glutamate and glutamine. So both are increased, and they also can feed from the astrocyte to the neurons. So this is actually important in terms of neurotransmission. I can also so there's, I mean, one thing that can change also is ketone bodies, because of the enhanced fatty acid oxidation that happens in APBD. But then you have actually, so this could be actually lowered by the treatment because you have less fatty acid oxidation. And this is speculative, I realize that, but possibly we didn't test for ketosis just because at the time we did targeted analysis and we didn't have ketone bodies as in internal standards. But now when we are what we're proposing now, and now we have also the capacity to do it, when we do untargeted analysis, hopefully we'll see what happens with the ketone bodies. So off the top of my head, I think these are like 3 directions that I can envisage that could be modified in terms of the metabolomics. In terms of the what we call phenotype, which cell features are going to be modified, we also know, because I mean, GHF21, we know its mechanism of action, we know its target, it has been very well characterized. So we know that there is enhanced lysosomal activity, lysosomes acidify, autophagic flux increased, and so I can envision, can relate it, as I mentioned, to metabolic change in the pathway, which is enhancement of the provision of energy substrate to the mitochondria. For instance, polyamines. Putrescine, which is polyamines which are degraded in the lysosome and are provided to mitochondria. This is one group of molecules that I can predict will be increased. And also we know, this is something that we know already about the cell features. So mitochondrial activity is enhanced. There is repolarization. They are depolarized in the mitochondria, depolarized in the patients. They can be repolarized. Lysosomes become smaller. These are the main cellular features. And also, of course, we know it from an unpublished work. It's now in bioarchives from Gilkey disease in GSD1A, that there's a tremendous change tremendous epigenetic change. Okay, there is an increase because there is an increase in electron transport in the mitochondria. There's a higher production of NAD plus, which is an activator of SIRT1, and therefore you're going to have major epigenetic changes as a result of this. So yes, I mean,
I have a short follow-up to that will actually lead to my question from the other group. Do you think that these findings will be most relevant from blood testing in people, or will it have to be CSF testing, or will it be a combination of both to really know where these changes are happening?
So yes, I mean, so far we did only blood test. CSF is more problematic. Not all patients are available for CSF, and one of the purposes here is actually to do the CSF and do exactly what you're asking about. I mean, just do the correlation, and then we can, from the mouse data, perhaps we can predict if blood tests are sufficient, if they correlate with the data we get from the CSF or not. If not, then we have to think about some patients. It's easy to get CSF, but from what I know from my interaction with the clinicians, with the neurologists, it's not trivial. On the paper accepts everything, but it's not trivial. So if we can, if as you know, I'm sure, but if we can, if blood and urine are can provide enough information, at least enough biomarker information, I would opt for them.
Great. There was a question submitted that I think also kind of builds from this for Dr. Koch and Dr. Jones. So the question that was submitted is, were there any places in the brain and spinal cord biopsy results that show high skew polyglucose antibodies? And I think this is a great question sort of in the context of this really precious and generous material that you have to study and help answer the questions and line up with the animal model to really build on our understanding. Does it matter where we look and test from?
I'll let Dr. Jones take it if that's okay, because I don't think I did a great enough job in the presentation of also explaining how critical she was in actually doing the research autopsies and directing to make sure that we get as much tissue as possible and from all the important regions. So if that's okay with you, Dr. Jones.
Oh, of course, yeah. So some of the brain regions, what we've tried to do is we've tried to really do comprehensive sampling. These patients have been so generous, and their families have been so generous to help us better understand this disease process. And so we wanted to make sure that we have sampled brain regions extensively and followed up with staining in all of those brain regions, treating them equally so that we can try to understand what's going on. And as Dr. Koch said, our findings are still preliminary. We haven't finished all of the analyses, but some of the brain regions that we're seeing a lot of polyglucosan include the hippocampus, the cerebellum. We are seeing, to correlate with the imaging findings that are noted in patients, the concern for leukodystrophy, we are seeing in both of the brains, we're seeing these regions where there's rarefaction of subcortical white matter. So this is, the white matter in the brain showing these areas that look like, in some circumstances, we could call them infarcts. I don't think they're really infarcts. They're more in the neurodegenerative realm of what we see with other leukodystrophies, but there's basically myelin is gone. The axons are rarefied, and there's a lot of gliosis. And these regions are oftentimes in, near the ventricles of the brain, which is why I think some patients get misdiagnosed with multiple sclerosis, because that is a classic region for multiple sclerosis lesions. So that is something that has been interesting to just see firsthand.
That's great. We are looking forward to our better understanding of where which areas are affected and in what ways to kind of move forward. And to kind of just one more step in that direction is I suspect that it will be different for everyone given how heterogeneous people can develop symptoms. Although the white matter changes may be pretty similar. We'll see. We'll see. That's my own prediction. Okay. So another question I have is for Dr. Kaklon. Do these inhibitors, Guaiacol, GHF 201, 205, work in other GSDs like Pompe and Lafora?
Yes, definitely. So, okay, we have many results. Some are published, some are not. For Pompe, we do have many positive effects of GHF201, which is, it's not an inhibitor, it's actually an activator of lysosome activity. There is mTOR involvement, which is improved, autophagy, of course, motor components, glycogen was reduced by GHF201, and also we looked at insulin resistance. We did a lot of work. It's not published yet, but I can definitely say that the GHF201 is a preferred drug for Pompe, and we did try combinations. GHF201 expectedly, I would say, as a lysosomal activator, it works for Pompe. For Cori disease, so GSD3, we also published work with GHF201. And again, we see definite improvement in, for instance, in myofibrillar dissociation. So this part of the muscle is improved. Also, in terms of, we also did metabolic cages to look at in vivo metabolism on the own animal, and we saw an improvement in the respiratory quotient. So in other words, they use more carbohydrates than fats, which makes sense with the improved degradation of glycogen. We also have a lot of data on mucopolysaccharidosis. We have, as I mentioned, some positive data in pancreatitis in GSD1A, reduction in liver glycogen, increase in liver glucose we did in the mouse, and in cells, as I said, there is a vast change of acetylation and methylation of the genome, and we looked also at the exposed regions of the chromatin, TACSEC, transcriptomics. We've done a huge body of work, and it's about to be published, but I won't go into too many details, but I can say that there is definitely not only an effect in the animal level, but also if you looked at the transcriptomics, for instance, we have expression of lysosomal biogenesis proteins, mitochondrial biogenesis pathways. So all these things work together, and I think we have very interesting results. On these, the combinations, as you know, it's new, so we don't have many results, but with GHF-205 in APBD, we do have interesting results, nociception, as I mentioned, and also the other parameters we looked at in APBD models. So yes, to answer, a short answer is yes. It did that in many other models.
Great. Another question for Dr. Koch and Dr. Jones. Based on what we know from autopsy for GSD4 in childhood onset and adult onset APBD, do you have some insights on potential shared pathways for biomarker identification and treatment opportunities for these two disorders and how that will come out of these studies?
I love how you give us the really hard question.
Yeah, no, straightforward, no problem.
I think I can take this one. I think there's still a lot for us to really know. I'll speak on our natural history study tomorrow live, but we're still really understanding the differences and also similarities in the tissue involvement between the children and the adults. We don't understand at this point why adults seemingly do not have any symptoms up until middle to late adulthood. Or if there is underlying tissue involvement, that's just like kind of a slow burn that eventually catches up and causes symptoms. But from what I can tell you from the autopsy and some of the published autopsies from children, as well as just tissue biopsies in children, there's a lot of tissue involvement that's very similar. So in the intestines, just for an example, and in other tissues as well. So not talking about the CNS because we don't have samples from healthy children from the CNS. Or children that are alive. But I think there's a lot more overlap than we are necessarily acknowledging. So that could help if we're talking about a treatment that could be impacting the whole body and not necessarily just targeted to the CNS. But I think that's something we'll still be learning, especially with the CNS, how there are similarities between children and adults. But I think that's a harder one to tackle, just given that we don't have many autopsies from children.
Right. There was a question that was in the chat that I think was answered, but maybe if Dr. Kaplan wants to just summarize it, about the guaiacol as a comparable structure to guaifenesin.
Yes, it's an ether also. It's an ether of guaifenesin is an ether of guaiacol. I think I answered it, and also someone else answered it.
Just to summarize it, if anybody didn't get to see that. Okay. All right, so I have another hard question for you while I read something that it just showed up here. So this one is for anyone who wants to answer. So in the current stage, we're in medicine, we're coming up with this precision medicine approach. Do you think that the biomarkers or these approaches will be variant dependent in APBD, or will one approach work for everyone? Anybody who wants to take that? It's a very easy, straightforward question.
I would guess they would be variant dependent. So when you say variant, you talk about the exact mutation or the background, or both?
I guess both, actually.
Yes. So I mean, just take, for example, the lipid fat oxidation status of the patient. It can make a tremendous difference, you know, on the metabolomic biomarker and fatty acid oxidation in general. So I think this is why we have to have, I think, multivalent biomarkers. Each one will address a different point, and then you have integrative biomarkers like the what I meant, the metabolomics and spatial metabolomics in the brain. So which areas in the brain has which metabolites and which pathways are modified in the hippocampus as compared to the cortex, for instance. So yes, I think we are going. This is where we're aiming at precision medicine, and the more biomarkers we have, they will be more precise. For instance, the neurofilament, the big advantage is that it is systemic, and you can see in the blood what goes on in the neurons. However, it only gives you 1 point. Okay, neurodegeneration in general, it doesn't go into details. And I think having both metabolomics data plus combined with cellular features or expression, transcriptomics, proteomics, and integrating them is really going to not only to provide more precise picture of each specific patient, but also of the therapy. Okay, because as I mentioned, if you look at the relations with after the therapy and you compare it just to vehicle treatment, you can find many new targets, and you can have more targets to select and actually adapt them or to each specific patient. So this is why I think the biomarker or the variability of biomarkers that we have now is so important, just for that, for precision medicine.
I'm going to add in as well with I'll just take the side of the comparing children and adults as well. I think also how much the biomarker represents the severity of the disease versus just generally if GSD4 or APBD are present, because that's something that we've been curious about also with children, of comparing some of these biomarkers like neurofilament to adults. And I think, like people have acknowledged with neurofilament, there are pros and cons, but that is something that we're trying to see. Okay, when we talk about underlying disease involvement, again, is that biomarker still helpful even if that patient doesn't show overt disease yet, or symptomatic in that way? So that doesn't really answer a question, but I just wanted to put that point out there as well as people are investigating those biomarkers.
And something that is now something that's clinically available that we can order on people to follow neurofilament light. So, you know, within your natural history study, it will be really important information to collect. So thank you for doing that.
I'll hint at it tomorrow.
we are doing neurofilament in patients as well, you know, before and after doing treatment. So we can add to that.
Combine our efforts.
Amazing. I see, I think we maybe have time for 1 or 2 more questions, depending on this next, not very straightforward, but great question. So I want to share this from Rob Schreier. What is the path you see from your excellent research to practical treatments of APBD patients? Have you documented the steps through the initial compassionate treatments? And are there areas where that path can be accelerated? I mean, again, I think these are things we're going to talk about over the course of this conference as well. But of course, I'll end it by saying, again, appreciate your research very much, because of course that's what we all do as a community, and we're happy to celebrate it today. But how do we get there?
Yeah, so I mean, actually, there's another session on that, as you know, with our CEO. But I mean, I think what Rob is, if I understand it correctly, is getting at is actually combination treatment also for patients. I think, and then you have actually, so okay, GHF-201 can degrade the glycogen. What if you have less glycogen to begin with? Will it be more effective? Okay, so if you give glycol before GHF-201, will the therapy be more effective? So I think things along these lines could definitely be considered, and this would be you know, and this would be the next steps. If I may just digress a little bit from APBD, when we have like pediatric disorders like that Becca is familiar with more than me, like Pompe or MPS I, we have ERT, and this is the gold standard. So you cannot, patients have to get the ERT, so you can only have combo treatments. Okay, you can only have add-on. You cannot replace the ERT. And so there we have actually a path which is dictated to us by the regulatory authorities. So however, sometimes we have to think about it. What happens if the ERT doesn't work? At which cases can we discontinue it? Can we replace it with the previously add-on therapy, which is now single therapy? So this is I think one of the questions I would ask. But I think I think combo therapy, especially when ERT is exhausted, you cannot get more from it, could be really a nice therapeutic path. So because I mean, and the thing, and I can see, I mean, we have bad results as I mentioned in mucopolysaccharidosis models that show actually that there is at least an additive effect. So if you treat with ERT plus something like GHF201, or in the case of mucopolysaccharidosis, catepsin inhibitors or whatever. So I think from this basic research, we can see how to add on to existing treatments or how to combine if there is no treatment.
Well, thank you so much, everyone here, for your important work. And we look forward to your future results and really appreciate what you shared today.
Sure.